Next, I'd like to welcome John Marshall. Dr. John Marshall is a professor of medical science at Brown University, and he's going to talk to us about the progress in his compound for BDNF signaling in Angelman syndrome. Impact the next steps.
Thanks, Allison. Thanks to FAST, thanks to Dr. Weber for inviting me. So when the brain forms new memories or we learn a new task, the brain does this by encoding the information by modifying synaptic contacts. And a growth factor that's really important in this mechanism is a growth factor called brain-derived neurotrophic factor. So today I'm going to tell you a little bit about brain-derived neurotrophic factor. And we found using the Angelman mouse model that Dr. Baudet talked about, learning deficits in the mouse model result from decreased activity of this growth factor, brain-derived neurotrophic factor. And we've been developing a new drug called SYN3 that can actually compensate for the loss of UB3A and restore learning in these mice. So this slide, it's hard for me to point, but this slide shows, synaptic connections. BDNF is really important for the strengthening of synaptic connections. When you have an impulse from one nerve, it passes down this process called axon. At the terminal, it causes the release of a transmitter, in this case glutamate, which is the amino acid, is an excitatory neurotransmitter, and it will excite the second neuron. And at that contact point, that's the synapse. And that's where you can either strengthen or weaken synapses, and brain-derived neurotrophic factor plays a really important role in that. So if you look at that cartoon on the left, when you get electrical activity or an impulse from one nerve, it causes the release of glutamate from synaptic vesicles. It's dumped into this cleft. It diffuses across where it interacts with glutamate receptors. They have the names AMPA and MJ receptors based on their pharmacology. But it will bind to those receptors, open them, and if there's a strong enough signal, it will activate that second neuron. But sometimes that doesn't happen. So BDNF, when you have activity, BDNF is released. It binds to this receptor called the TrkB receptor, and that recruits a protein called PSC95, and that's our drug target. And that's really critical for activating a number of enzymes that are critical in strengthening the synapse. Some of those enzymes include CAMK2, an enzyme that Dr. Weber worked on. CAMK2, one of its roles is to stabilize more glutamate receptors at the synapse. So you have more receptors, you're going to get a stronger signal and a better chance of transmitting that signal. And CAMK2 will also activate other enzymes. One is called mTOR, and that's really important in lots of things like forming the branches of neurons. You can see I have an image of a neuron. This is from the hippocampus, and you can see it has these branches like a tree. The more branches, the better. In Angelman syndrome, you have fewer branches, and along those branches, that's where you form the synaptic contacts. And you can see, perhaps, those red dots. We've stained for our protein PS95, and that's where you have the synaptic contacts. And I'm generalizing, but the more branching, the more synaptic contacts, the better. Although that's not always the case. Sometimes you can have too many synaptic contacts, and that can lead to autism. And you can also have fewer synaptic contacts, and that will lead to Angelman syndrome. Yes. So this shows a little bit more detail about some of the enzymes that I talked about. I mentioned BDNF binds to the receptor. It activates a number of enzymes, ending, for example, in mTOR, which inhibits the process that Dr. Huang mentioned called autophagy. So we have a drug that actually slows down autophagy. I'm not going to talk about that today. And as I mentioned, it activates CAMK2. But in the Angelman mouse, those enzymes are not as active. And you can see that if you look at this is called a Western blot. At the top on the left, you see strong bands. That shows a lot of activity. Whereas in the Angelman brain, and this is from the hippocampus, you see a fainter band for that phospho-mTOR. That tells us that mTOR is not as active. And similarly, if you look down, if you look at CAMK2, another really important enzyme, its activity is also decreased. So what we want to do is kind of restore the activity of these enzymes. The problem with Angelman syndrome is UB3A does a lot of different things. One of the things it does, it regulates transcription. So it actually regulates the expression of BDNF itself. So in Angelman syndrome, you have decreased levels of BDNF for a start. Also, it can inhibit other transcription of other proteins. One of the proteins it inhibits is a protein called ARC. And so when you don't have UB3A, you have too much ARC, and we find that ARC binds the PS95 protein and prevents it from performing its proper job, which is enhancing TrkB signaling. So we've developed a drug called SYN3 that binds to PS95, and so it kicks out ARC, and now you restore normal activity of these enzymes. So now you can see on the top right, now you've got a stronger band for phospho-mTOR is more activity, and similarly you have more activity of CAMK2. So now this drug can restore the normal activity of these enzymes. I'm just going to show you this. And this is important for restoring normal dendritic branching, restoring normal synaptic activity. So it's not important, but it's a peptomimetic type drug. We've been developing a number of these. One is called SYN3, one is called DSYN3, which is a more stable drug. That's actually our best drug so far. And these compounds, they bind to the drug target. The target is PS95. It has all these domains. It has PDZ domains, an SH3, a GUC domain. But what this drug does, it binds to the PDZ3 domain and also to that alpha helix. So it binds in two places. So it binds tightly to two places. That gives it higher affinity. That compares to our older drug where it only binds to one place. And so our older drug has a lot lower affinity. So this new drug, we can give at a 10-fold lower dose, and it's still really, really effective. And it has great specificity because it's, has to bind to these two sites. So as I mentioned, this drug binds to the PS95. It kicks out ARC. ARC is important for a lot of things, but if you have too much, it's not good. And then when BDNF binds to the receptor, PS95 gets recruited, and that helps to restore normal signaling. Okay? So this shows we've injected this drug into our Angelman mice, and you can see when we stain for neurons, and this is called a pyramidal cell in the hippocampus. On the left is from a wild-type mouse. You can see really nice branching, it's shown in green. You see beautiful branching. In the middle, you've got an Angelman mouse. You can see that there's much fewer branches. Actually, a little bit like depression. We do a lot of work in depression. You get the same thing. Your branches retract, but we can fix that. And we have drugs for depression that help the branches grow back. In Angelman syndrome, when we give SYN3, our branches grow back. And those branches, as I mentioned, are the place where you form the synapses. And you can see on the right, at the top, we're looking at these little protrusions from the branches. They're called dendritic spines. And those are the places where you form your actual contacts. In the Angelman mouse, you see fewer of those protrusions, so you've got fewer synapses. But when you give back our compound SYN3, you restore those protrusions. It's pretty amazing. And presumably you're restoring synaptic contacts. Okay, but how do you show that? So we perform electrophysiological studies, and on the top left you can see a cartoon of the human brain, and you also have a cartoon of the mouse brain. And the human brain is a little bit different. You can see the convolutions in the cortex, which are really important. But really, at the microscopic level, there's not much difference between a mouse and a human brain. The neurons kind of look the same. We study a part of the brain called the hippocampus, which you can see outlined. There's, you can see the human hippocampus, you can see the mouse hippocampus, and that hippocampus is really important in forming new memories. You take in information like a name, and you can either remember it, or you can then transfer that information into your long-term memory. If you damage the hippocampus, you can no longer make new memories. So the hippocampus is really important in temporarily storing information, transferring that information into new memories, called consolidation. So we wanted to look, does our drug help restore the strengthening of these synapses? And we record, we put an electrode into the mouse hippocampus, we record activity, and if you stimulate in the right way, you get a response. And unfortunately, in the Angelman mouse, when you stimulate, and hopefully you're trying to form a new memory, it doesn't get any bigger, it stays the same size. That's the problem. If you stimulate in the right way in a wild-type mouse, you get a stronger response. That's referred to long-term potentiation. That's the assay that we use to test our drug. And so when we inject our drug into the Angelman mouse, now when you stimulate, you get a bigger response, and that response is maintained for a long time. That's called LTP, long-term potentiation. And so our drug does that, and that sort of bodes very well in terms of, okay, is it going to help in terms of memory in these mice? The other thing I want to point out is on the lower right, we can inject our drug, and we can wait 3 or 4 days, and then record, and we still get LTP. So these drugs are long-acting, which is great. So if you can imagine giving the drug to a patient, you might only have to inject it once a week, for example. So, like insulin, or even better than insulin. One slight difference between a mouse brain and a human brain you might notice, the mouse brain is a lot smaller. I was going to make a joke about Trump, but I got so depressed I thought I wouldn't make that joke. Anyway, but what's cool about the human brain is it's very plastic. It has a lot of adaptability. So we think that our drugs are going to work even better in the human brain because the human brain is so amazing at making changes and forming new connections. So we think that these drugs will work even better. So we've tested these compounds in terms of different learning paradigms, in terms of cognition. These studies were done in collaboration with Jill Silverman as part of a FAST funding collaboration. Jill is one of the world's experts at doing these learning paradigms in animals. And in this particular test, the mice get to explore two similar objects. And then after a while, we trick them, we kind of remove one of the similar objects, and we add a novel object. And a wild-type mouse will say, hey, look, I remember that familiar object. I'm going to start exploring that novel object. Unfortunately, the Angelman mice don't do that. They don't remember that they previously experienced the familiar object, and they explore both similarly. When we give our drug, now the Angelman mice remember the familiar object, and they spend more time checking out the novel object. So that shows that we are improving cognition in these mice, at least. We've also looked at motor coordination, of course. Do these compounds improve motor coordination? Our DSIN3 compound, the more stable compound, does seem to have a very significant effect on motor. If you look in panel B, that middle curve is our DSIN3. It improves motor learning. It's not as good as the wild type, but we've also know from experience if we treat the animals when they're younger, these are probably 3-month-old animals, which is the equivalent to, 20, 30-year-old human. If we treated the animals when they're much younger, we can restore their coordination similar to wild-type mice. The other surprising thing was in terms of seizures. These drugs really have a lot of efficacy in terms of reducing seizures. So in these mice, we inject a drug called pentylene tetrazole, and that will induce seizures in AS mice very rapidly, compared to, say, a wild-type mouse. And then when you inject our compounds, we can increase the latency to get seizures. So we actually takes longer to generate these seizures, which is really could be really useful. We're not really, we didn't set out to develop a drug for seizures. We're interested in improving cognition in humans. That's what I wanted to improve memory, improve speech, etc. But it's really nice to see that the drugs also have this anti-seizure activity. And this is an Angelman mice, but we also checked out just wild-type mice. It actually helps decrease seizure activity in wild-type mice as well. So in terms of the next steps, I've talked about we have our drug target. We've done a lot of medicinal chemistry. We still want to do a lot more medicinal chemistry. We're actually designing some new drugs that have even higher affinity. We've done the in vivo studies. The next step is trying to get towards clinical trials, and that will require, obviously, a lot of safety work. We want to make sure that these drugs are safe, look at toxicology, that type of thing. So that's kind of where we're going next, hopefully in the next year or so. I'll do it for time. So that's it. Finally, I just want to acknowledge the people who did the work, particularly the Silverman lab. Jill Silverman is, as I mentioned, really did most of all the behavior work, and Emily Hui did that. It's just amazing work. I have a collaboration with Eric Levine. He's going to talk later. Eric does amazing work looking at the electrical activity of neurons, and particularly when UB3A is absent, so you might hear some of that. I have a collaboration with Michael, I can never pronounce his name, Cao, he was a postdoc of mine. Now he's a professor in China, in Suzhou. We're collaborating on some of those dendritic spine studies and some other signaling studies. My lab, of course, on the left. Menja did all the electrophysiology. Alvin's lab, Alvin Huang, who just spoke before me, we have a very amazing collaboration, and particularly Xin Yang. He helped us a lot with the autophagy work that's just been published, actually, and some of the iPSC work. And some of the compounds we're discovering are very useful for other disorders like hypoxic ischemia and cerebral palsy. And we're also getting some really nice data in Alzheimer's models. So with that, I'd like to thank everybody. That's it.
Great. Thank you so much. I think we'll reserve questions just to stay on time for the panel. So we'll get back on the panel and get all the questions. But that's it's super exciting to realize that there's, very complicated, expensive, and challenging permanent gene therapies and cell therapies and antisense oligonucleotides. And when you think about the downstream targets and how much work UBE3A does and the impact UBE3A has, and you realize understanding the mechanism at a deep level at the synapse is really challenging. And those last two talks really show you how they're working really hard to try to figure it out and understand the best options for downstream targets, which is really exciting. So thank you both. The team at Brown has been amazing to work with, and we've been working closely with them for a couple years.